Hey, welcome to the Biochemistry Academy. We began a serial course on nucleic acid chemistry, or simply the chemistry of nucleic acid. And in our last video, we introduced what nucleic acids are and briefly introduced what DNA and RNA are. Well, in this video, we'll focus more on DNA. We are going to understand the structure of DNA, the types of DNA, and the properties of DNA. Now, DNA is a polymer consisting of small repeating subunits called deoxyribonucleotides, which are the monomers that build up to form this DNA. DNA by its structure is double helix, and this structure was proposed by James Watson and Francis Crick. Now, if we check the nucleotides in this our structure here, they are joined together by a 3' to 5' phosphodiester bond. Now, it is formed between the 3' carbon of this nucleotide to the 5' carbon of the other nucleotide. And if you notice, at this position, we have the 5' end and the 3' end. In the 5' end, it means that the terminal is not linked to any other nucleotide. And at the 3' end, it means that the terminal is not also linked to any other nucleotide. Now remember, we said that the structure of DNA is double helix, and it was proposed by Watson and Crick. Now we are going to understand the Watson and Crick model of DNA. DNA is a right-handed double helix and also having two antiparallel strands, meaning that one strand runs in a 3' to 5' direction and the other strand runs in a 5' to 3' direction. To this, one can say that both run in an opposite direction. Plus, both of these strands are complementary to each other. Now, what do we mean by this? We mean that A pairs with T and G pairs with C. A here is adenine, T is thymine, G is guanine, and C is cytosine. We talked about it more in our previous video. So now, the amount of A is equal to T and the amount of G is equal to C, meaning that the total purine is equal to pyrimidine, which proves the Chagall's rule. The two strands are held together by hydrogen bond that are developed between the purine and the pyrimidine of opposite strands. Adenine always binds with thymine with two hydrogen bond, while guanine always binds with cytosine with 3 hydrogen bond. Oftentimes, students are asked, why does purine pair or bond with pyrimidine? Now, the reason is one, purines always binds with pyrimidine in DNA due to their complementary shapes, sizes, which facilitates effective hydrogen bonding. Now, purines like adenine and guanine have two-ring structure, while pyrimidine, such as thymine and cytosine, have a single ring structure. Now, this bearing maintains a consistent width of the DNA helix, which is essential for structural stability. Both strands of DNA can act as a sense and antisense strands. The sense strands of DNA is also known as the coding strand. It has the same sequence as the mRNA and it runs in the 5' to 3' direction. It is not transcribed into RNA, but it serves as a reference for the mRNA sequence. The antisense strand is also known as the template strand, and it runs in the 3' to 5' direction, and it is complementary to the sense strand. Now, it is the strand that is responsible for transcribing into mRNA, during protein synthesis. Understanding these strands is crucial for molecular biology, such as in gene expression analysis. Next, we'll talk about the types or the forms of DNA, comparing their structural features. DNA takes different forms and it can be classified as aDNA, B, C, D, and zDNA. But the most common are the aDNA, bDNA, and Z form of DNA. Now let's begin with the A form of DNA. The A form of DNA is a right-handed helix. It is present in a dehydrating environment in the presence of sodium ion, potassium ion. Now the number of base pairs in each turn is 11 base pairs. Now there are two grooves, which are the major and the minor groove. In the major groove, it is narrow and deep, but in the minor groove, it is wide and shallow. Now, the next form is the B form of DNA. Now, the B form of DNA is a right-handed helix, which is present in a very high relative humid environment and in low concentration of ions. Now, each turn contains 10 base pairs. Now, in the structure of the B form of DNA, there is the major and the minor groove. In the major groove, it is wide and deep, while in the minor groove, it is narrow and deep. Next, we'll talk about the Z form of DNA. Now, the Z form of DNA is a left-handed helix. Now, they have a zigzag sugar phosphate backbone running in an antiparallel direction. Now, the reason for this zigzag form is because of the sequence in it and the alternating purine to pyrimidine on it. Now, each turn is 12 base pairs, so it is longer than the A and the B form of DNA. Now, in the major groove of the zDNA, it is flat, and in the minor groove, it is narrow and deep. Now, we'll understand the properties of DNA. DNA undergoes denaturation, regeneration, hyperchromicity, decreased viscosity, and DNA hybridization. Now we'll talk about the first property, which is denaturation. Now it is the unwinding and the separation of two strands of DNA. The covalent bonds are not broken. Only the weak bonds like the hydrogen bond, the electrostatic bond, and the hydrophobic interactions are broken. Now we say that denaturation involves the unwinding of the two strands of DNA. Next is renaturation, which is also known as annealing. Now, when the temperature is cooled, then it brings about the reassociation of the separated DNA strands to form a double-stranded DNA structure. Now, this denaturation and regeneration of DNA is used in polymerase chain reaction. Next is hyperchromicity. Now, the DNA, which is denatured, shows increased absorption of light at 260 nanometer. Now, in the separated strand, purine and pyrimidine base are exposed to more light. This property is used to estimate the purity of DNA. Now, the next property of DNA is decreased viscosity. Now, it occurs due to separated strands, which are more flexible than the rigid rod-like double-stranded DNA structure. The next property of DNA is DNA hybridization. Now, the base pairing of two complementary DNA strands form two different species to form a duplex DNA. Please don't forget to like, share, and subscribe to the Biochemistry Academy for more content like this. Arigato.